Hi everybody, I hope you're all enjoying your day off. It's nice weather that we're having. For those of you that still have power, I thought I'd prepare this video lecture today. It's just going to be my voice over the slides, and hopefully that's enough. If you have any questions about anything in the lecture, feel free to just email me, and I'll get back to you as soon as I can. So that being said, today we're going to talk about mutagenesis, so making mutations in proteins. So I'd like to start with a case study. So this is a jellyfish, Aquaria victoria, and what makes it special is that it has a fluorescent protein. Specifically, it's a green fluorescent protein. The acronym for that is GFP, which you might have heard of before. The GFP allows the jellyfish to give off light, fluorescent light, that attracts different prey in. So fish will swim up to this guy thinking he's interesting, and then he'll swiftly kill the fish. So how does he do that? Well, he has a specially tuned system of proteins within him that starts with this protein called aquarin. Now, aquarin is activated by calcium. So the jellyfish can control the release of calcium, and that binds to aquarin to initiate a chemical reaction that releases blue light. That blue light is then transmitted to GFP, our fluorescent protein, and since GFP is fluorescent, what that means is that it absorbs one color of light and emits another color. So in this case, it absorbs blue light and emits green light, as you can see in the picture here. So even though there's blue light coming from the aquarin, you don't see that because it's all turned into green light by the GFP. This green light can travel farther through the water than the blue light. That's why the jellyfish bothers to have this second fluorescent protein in the system. Now the nice thing about GFP is that we don't really need aquarin to excite it. We can actually just use UV light. Actually, under normal white light, you'll still see some green color coming from it. So it's a very powerful fluorescent protein. So we can use UV light in the lab to get green fluorescence out of it. And we can use this in cells to label specific things. So if we make a fusion protein, let's say we fuse the GFP to an antibody, we can put that antibody GFP fusion inside cells and see where things are localized. So this picture on the right here is showing where this ERD2 protein is localized within the Golgi apparatus in a plant cell. So all those little blobs you see are the ERD2 protein. So something we couldn't see before, now we can see because it fluoresces green. And GFP a few years ago won the Nobel Prize because it's so widely used. So it's a great example to talk about today. All right, so let's begin our discussion of GFP by looking at the structure. It's a pretty simple protein, not too big. It's got a barrel shape, and the barrel is made out of these beta sheets. So remember your secondary structures for peptides. You can have alpha helices or beta barrels. In GFP, you have about 11 beta barrels, and they all are wrapped around this central fluorophore. It's labeled chromophore here. That's what some people call it. And the chromophore, or the fluorophore, is where the light chemistry takes place. So that's where the blue light shines in, and then these amino acids absorb that and emit it as green light. So the fluorophore itself is actually a special structure that's made from fusing 3 different amino acids. So these are amino acids 65 to 67 in the peptide chain. And you can see in the bottom left, in the top left, there's serine, tyrosine, and glycine. So starting out, we just have these amino acid residues, just like any other protein. But the special thing about this tripeptide, these 3 amino acids, is that if you put them together, they will naturally cyclize. So you can see in the first step here, we have the serine coming up to meet the glycine, and we get this 5-member ring. And then we have a dehydration step, remove some hydrogen, remove some water, and then an oxidation step to give us our final fluorophore down there in the lower left. And that's the one that's highlighted in green. So you can see we've pretty much just fused the peptide backbone, and now we have this structure that is fluorescent. So the beginning tripeptide up in the top left does not fluoresce. But when we link that backbone, we get this 2-ring structure that actually does fluoresce. And you'll see this type of structure in a lot of other fluorescent molecules. So it's kind of amazing that biology can do this automatically and naturally. Now the interesting thing about the fluorophore is that if we change amino acids around it or within it, it influences how the fluorophore absorbs and emits light. Specifically, remember, we have those amino acids, the 65 through 67. You can see in the middle of the diagram here, if we change the serine at position 65 to a glycine, that's the yellow oval you see, it changes GFP from a green fluorescent protein to a yellow fluorescent protein. So by changing that one amino acid there, we can actually get a different color out of the same protein. And you can see there's many other mutations in this diagram, and they all have different effects. For instance, you can see at the 66 position, if we change that Y to a W, we go from green to blue fluorescent protein. And there's actually two different types of blue fluorescent protein. The light blue is called cyan fluorescent, and the dark blue is actually just called blue fluorescent. There's also some green ovals you'll see all over the diagram. For instance, F99S on the right side. Whenever you see a green oval, those are mutations that actually make the normal wild-type GFP even brighter. So we have versions of GFP called enhanced GFPs, or eGFPs, that are much brighter. They're easier to see at low concentrations. And then finally, there's also a few gray mutations. These are mutations that make all of the different fluorescent proteins brighter or more stable. So all good mutations, those ones in gray. If you want a really bright blue one, then you would make all of the blue circle mutations. If you want a really bright yellow one, you would go for all the yellows. So what are these mutations doing? Well, if you look at the fluorophore itself, first of all, shown in the lower left is the fluorophore for eGFP. You can see the serine, the tyrosine, and the glycine. Now if we go to the top left, you'll see blue fluorescent protein. We have one amino acid difference. Instead of a tyrosine, now we have a histine. So that histidine, you'll notice, is a 5-member ring instead of a 6-member ring. And that lone change is enough to change the color of that protein from green to blue. If you go to the top right, now we have that cyan fluorescent protein. It's just a lighter blue color. What we've done there is changed both the tyrosine to a tryptophan and the serine to a threonine, and that changes the color completely. So here we have a larger ring structure in CFP, and it gives you that lighter blue color. In the bottom right, we have something else altogether. We've changed the serine to a glycine, which doesn't have much of an effect by itself. But when we change a distant amino acid, here we have tyrosine 203 is the mutation here. That ring sticks into the fluorophore and is able to change its fluorescence. So there, just by sticking that extra ring in there, we go from green fluorescent protein in the lower left to yellow fluorescent protein in the bottom right. So all kinds of subtle changes can make can have a very big influence on the color that this protein emits. And we've made a lot of different mutations for GFP. So everything you see here is some kind of derivative of the green fluorescent protein from Aquaria. And you can see we have blues, greens, yellows, pinks, and reds. Lots of different options here. And you can do some pretty interesting stuff with these different mutants. So the benefit to these mutants is that you can show different things at different times, or different things at the same time. In the lower left, we have a picture of a brain bone. So this is actually a picture of a mouse brain that's been stained with these different proteins, and they bind to different parts of the neurons and such. And so you can see different proteins and different neurons being activated at different times, and that's how you get the different colors there. On the right, we have E. coli. So the same bacteria that you'll be using in your experiments have been transformed with genes for these proteins, and you can see they fluoresce different colors. So somebody took an agar plate and made that little painting there, this little sunset. So how do we use all these mutants? So for instance, how do we make that brain bone? Well, you have to look at the the fluorescent properties of each mutant. And that's what's shown up here on the top left figure. So what you have on the first figure is the absorption, or what's called the excitation wavelengths for each protein. And you can see each of these proteins has a different maximum absorbance, or excitation. For instance, the enhanced blue fluorescent protein, which is shown in purple, absorbs at around, let's say, 380. Whereas our green fluorescent protein, GFP, which is shown in the light blue, absorbs around 475. So they absorb different wavelengths of light. And then on the right, it shows the wavelengths they emit. So the blue fluorescent protein, highlighted in blue, emits at around 450. Green fluorescent protein, highlighted in green, emits at around 500. So this is very useful because I can control the wavelengths of light coming into a sample. And I can also filter out the wavelengths coming off. So let's say I wanted to stain two different structures within a cell. If I use the same wavelengths for blue fluorescent protein and green fluorescent protein, what would come off would be a mixture of the colors. I wouldn't be able to discern these are the blue structures and these are the green structures. So in the bottom right, we see a bunch of different structures that are fluorescing blue and green. So if I combined A and B, I wouldn't be able to resolve the two. But by having these different mutants with different excitations and emissions, what I'm able to do is, for instance, in A, that upper left panel there, is shine a wavelength that's around 350 nanometers onto the sample, and it only excites the blue fluorescent protein. So I only see the blue. But if I just wanted to see the green structures, then I would shine a wavelength of, let's say, around 475 nanometers onto the sample. I won't see the blue anymore because my blue fluorescent protein doesn't absorb at that wavelength. However, I will see all of the green because the GFP absorbs that wavelength at 475 and emits at 500. So I can take these individual images and then overlap them later if I wish. And so that just allows me to pick out different structures within the cell. Very useful stuff. Alright, so I think we've established by now that mutants are very, very useful, and we can go much farther than just fluorescent proteins. You can take enzymes that are suboptimally active and increase their activity by orders of magnitude by making mutations. You can also increase stability of a bunch of different enzymes, make them a lot stronger than they used to be, make them withstand high temperatures, which can be very useful in industrial processes. So, if we know mutations are a good thing, how do we make them? Well, we're not going to use radioactive spiders or gamma radiation. No, those actually don't work. Well, gamma radiation might work, I don't know, I've never tried it personally. But what we're going to do, at least virtually today, is use PCR. So this is the same PCR concepts that we talked about during our last class, where you take a bit of DNA and then you copy it. And remember when we did PCR, we made modifications to the ends of the genes. We added restriction sites to them. Today, to make these mutations, we're actually going to make changes interior, on the inside or middle of our primers or our gene. So, let's say we have this sequence shown here, and we want to change the codon that's underlined there, that TAC. There's a lot of different things that we can change about this sequence. We can make a point mutation, so we can change that TAC to TTC, and that would probably be enough to change that from one residue to another. We can also make multiple mutations. Sometimes you need to change more than one letter to make a codon change for a different residue. So you can change one base pair, you can change 3. You can also insert base pairs. So here we have, instead of just TAC, now we have TAC TTC. So the sequence just got longer. And of course, in converse, you can also delete base pairs. So you can take out that amino acid altogether. Now the first two are a little bit easier than the second two, and we'll talk about that in a little bit. Okay, so mutagenesis, which is what we're doing, we're making mutations, is just like PCR. So the first step is making primers for the reaction. So we take our gene sequence here, and we split it up, just like we did for PCR. And what we're going to do now is make our primers. So our primers here are shown in green, and you start by making a complementary sequence. So again, we need a forward and a reverse primer. We need both of those. And our primers here are going to be a little bit longer. They're actually going to be around 20 to 40 base pairs. And our mutations are going to be right in the middle. So you see I have those highlighted in red. So we're going to change the C to an A on top and a G to a T on bottom there. And you want to make sure you're making the same mutation in both primers. And like I said, on either side of that mutation, we want 10 to 20 complementary base pairs. Now this is very important because those flanking regions there, those 10 to 20 base pairs, are going to be what actually holds the primer onto the DNA. We've created a weak spot here in our mutation, so that T doesn't want to bind to the G. So we have to have plenty of base pairs around it to force that primer to still bind. And you always, like I said, you always want to have that mutation in the middle of the primer. So some general guidelines for designing these primers, pretty straightforward. So like I said, you want to always order 2 primers. Make sure that they're reverse complements. So unlike PCR, our primers are going to be overlapping. They're going to have essentially the same sequence. Just one is going to be the reverse complement of the other. Your mutations should always be in the middle of those primers. And your primers should be around 25 to 45 base pairs. Now that's a pretty big range, so how do you decide where you need to be within that range? Well, if you have a high GC content in your primer or your gene, then you can use fewer base pairs. Remember, GCs have more hydrogen bonds, so they make a stronger, they anneal stronger than A's and T's would. So if you have lots of G's and C's, go ahead and use 25 base pairs. If you have A's and T's, you're probably going to end up closer to 45. If you decide to insert or delete bases that were not previously there, so if you're taking just ATT and extending it to ATT-CGG, adding those 3 different base pairs in there, then you're going to need more base pairs around it because that structure is going to be less stable. So if you're doing an insertion or a deletion and not just a point mutation, you're going to want to use 45 base pairs, a longer primer. Now, when you go to order these primers from a company, they're going to prompt you and ask you if you want your primers to be purified. And they're probably going to tell you that it's necessary, or it's going to highly increase the efficiency of your reaction. I'll tell you this, I've done mutagenesis several times. I've never paid for the extra purification, and it's always worked for me. The purification itself adds about $100 onto your primer cost. So we're talking about a quarter per base pair here. So primers that are 40 base pairs should only be about $10. You're going from a cost of $10 to $110 by purifying the primers. If you try your reaction and it doesn't work, maybe you go back to the company and you order the purified ones. But if you're doing it for the first time, you don't need it. All the purification does is when they make these primers, they come out as a mixture. So let's say you're making a 40 base pair primer. You're not going to get a homogeneous mixture of all 40 base pairs primers. Some of them are going to be 10, some are 20, some are 30, so on and so forth. So if you had the 10s and the 20s and the 30s, they're not going to work in your reaction. They're actually going to make it work less well. So if you purify them, then you only get the 40 base pair primers. So I'll leave that up to you if you want to do it or not. But yeah, here, this figure shows that exactly. So you don't want all the red, shorter primers, you just want the long green one. They use LCMS or PAGE to purify those, but it's expensive and usually unnecessary. So just a heads up there. You can calculate the annealing temperatures of your primers just like you did for PCR. You can use the same websites, but here instead of shooting for 40 to 55 degrees C for your annealing temperatures, we actually want to be up around 78. So we want a really, really stable primer duplex. And again, you want to make sure that both of those primers are about the same annealing temperature. They should both be around 78, not 180 at 170. You can calculate your annealing temperatures using this equation right here. And what we have melting temperature, Tm, on the left. You'll see percent GC, so that's how much of your sequence is a G or a C. N, that capital N in that fraction, is the total number of base pairs in your primer. And then percent mismatch is how many base pairs you're changing. So if you're just changing one base pair out of 50, that would be a 2% change. Now, this equation right here, as it stands, applies for point mutations, if you're just changing a T to an A or a CC to a GG, let's say. But if you're doing insertions or deletions, you modify this equation by saying that the percent mismatch equals 0. So that just defaults you to a higher annealing temperature that will help you deal with those mismatches in an insertion or deletion. Okay, so once you have your primers, you can go ahead and start with the PCR. So everything here is just about the same as with normal PCR. You still have Taq polymerase, you have nucleotides, a certain buffer, everything like that. And you start off with a template plasmid. So in cycle 0, all we have is the normal template. We don't have any mutations. When we go to the first cycle, remember we melt the DNA. So here we have single-stranded circle of DNA here. And then we lower the temperature and our primer anneals. And the mutation is shown there in the middle of the primer with a star. And DNA polymerization is going to happen in a 5' to 3' direction. So that chases around the circle. And here we go. Now we have a hybrid plasmid where one of the strands is mutated, the other is not. And notice that we have that nick right there on the 5' end of the primer. In cycle 2, we take that mutated part of the plasmid, and now we're going to use our reverse primer. So that reverse primer overlaps the original one, and again, DNA replication, 5' to 3', and there we go. So after 2 cycles, now for the first time, we have a plasmid that is fully mutated. But it's very important, you have to notice, we have these nicks. So the polymerase will make the copies of the DNA, but it won't seal the circles. Now this is why our primers are overlapping, is because if we do have these 2 nicks, at least the nicks aren't overlapping. So if the nicks were overlapping, we wouldn't have a circular structure. But since they are not, we still have the circular structure. It's just a little less stable than a normal plasmid. Now, since we have these problems with stability and these nicks, we actually use less cycles for the PCR. If you're doing a point mutation, you only use 12 cycles. If you're doing a multiple base pair mutation, use 16. For insertions or deletions, the efficiency of those reactions are even less. You go up to 18. Now remember, in PCR we use 30, or at least 25 cycles. Here, we don't really need to do that. So strike what I said earlier about this being less efficient, so let's use more cycles. Here, we don't need to make a billion copies of this plasmid. We only really need 1. Once we have that 1 mutated copy of the plasmid, we can put that in bacteria and let the bacteria amplify it. So there you go. Sorry about that. I misspoke. That's why we only use 12 cycles instead of the usual 25 to 30. The mutagenesis PCR routine is pretty similar to a normal PCR routine, but you'll notice a few different changes. In the initial melt step, that only has to be around 30 seconds. Unless, of course, you have high GC content, then use a lot more time. Then we have 12 to 18 cycles. And notice that our annealing temperature is a fixed 55 degrees C. All of our primers should have an annealing temperature much higher than that. So we only, any mutagenesis reaction you do, you'll set that annealing temperature to 55. And notice it's no longer 15 to 30 seconds, it's 1 minute. So we give the primers a little extra time to anneal, since they are kind of funny. They have those mutations in the middle. And then, just like any other PCR reaction, you extend at 68 degrees C for 1 minute per kilobase. At the end of that reaction, then, you have a final extension. So any plasmids or sequences that aren't fully replicated, you go back and finish those, and then you hold it for C. So pretty similar strategy. Now, once we've done the mutagenesis PCR, we're left with a mixture of products. We have the original template DNA still in there, which doesn't contain the mutation, and we have our new plasmid that is mutated. So, take a second here and think about it. How can we get rid of that original template? Is there anything different between the original template and the mutant plasmid? Well, I won't take too long here because I don't expect you to know the difference, but here it is. So that original template, what makes it special is that plasmid on the left came from a bacteria. It was synthesized in a cell. Now, in most of your living cells, they have enzymes that methylate the DNA. So they add methyl groups to the DNA backbone, or to the base pairs. And so the plasmid on the left has methyl groups sticking out from it. The plasmid on the right does not, because we made that in vitro. That was made outside of a cell, so there's absolutely no methyl groups on that mutant plasmid. And fortunately enough, there are restriction enzymes that degrade DNA that have methyl groups on them. So, for instance, here we're going to add DPN1, and this eats away any DNA that has methyl groups on it. And this just takes an hour at 37 degrees C. So we add the DPN1 to our mutagenesis reaction, and surely enough, the original template goes away, and we're left with only mutant plasmid. But we still have those pesky nicks, so we don't have a perfect plasmid just yet, and we probably have a pretty low yield. There's probably only a few of these molecules in the solution. So how do we, first of all, repair the DNA and amplify it? Well, like I said earlier, we're going to use bacteria. So bacteria will readily take up this plasmid in transformation, and that's a lab we'll do in a few weeks. I mean, if it doesn't snow on that day, let's hope. So if we just mix our mutated plasmid with E. coli, they take it up and they fix it. The bacteria has enzymes that will seal those nicks and make a lot of the plasmid. So every time the bacteria divides, it makes new plasmid. So there you go. We have two birds with one stone. We've repaired the plasmid and we've made a lot of it. Once you have a lot of the plasmid, you want to sequence it. So you want to send it off for DNA sequencing to make sure that this is the plasmid, this is the mutation that you were hoping to get. It doesn't always happen. Sometimes the original template can survive the DPN1 digestion, or sometimes your primers don't do exactly what they're supposed to. So you always want to check the final product of this transformation with DNA sequencing, and we'll talk more about that in subsequent lectures. Okay, so just to review. First step in mutagenesis is to do the PCR, and that's going to give you two products. It's going to be the original template and the mutated plasmid. Then you're going to add DP1 to digest the template, so that gets rid of the unmutated gene. And then finally, you take that nicked plasmid, put it into the E. coli, they fix it, and make a lot more of it for you. So, just in case you ever do this, first of all, when you're setting up your mutagenesis reaction, you want to use a very low amount of plasmid DNA. Right? You don't need a high amount, so only use 10 nanograms. You want to keep that pretty low. And then you want to add about 125 nanograms of your primers. Again, a pretty low amount. And then you can buy kits for this, kits for mutagenesis from a lot of different companies, and they'll give you a master mix that contains the nucleotides, the buffer, and the Taq polymerase. Now, since you're copying an entire plasmid, one key difference between this and PCR is that it's going to take several hours. So instead of copying a 1,000 kilobase gene, you're copying a 10,000 base pair gene. So it's essentially 10 times longer depending on your plasmid. So make sure you set aside a day, most of your day, to complete mutagenesis. And then when we add DPN1, Usually DPN1 comes with the mutagenesis kit. You just take the finished reaction from the PCR thermocycler, throw in a microliter of DPN1, and put in the water bath for an hour at 37. That gets rid of your template DNA pretty effectively there. And then finally, for transformation, again, most mutagenesis kits come with E. coli cells, and they're in little aliquots of 50 microliters. You just take a few microliters of your digest reaction, put it into with the cells, and let it go for about 2 hours. And you guys will see how that transformation reaction works next week, or in 2 weeks. But pretty simple. But it does, as you can see, take about a full day to do this whole reaction. Alright, so we just walked through the process for making a single mutation. What if you wanted to make 2 different mutations? So remember, we had the yellow fluorescent protein mutants had several different mutations, right? They had about 5 or 6 of them. So if you know the mutations you want to make, and there are several of them, how do you do that? Well, it's a pretty simple transition. First of all, you could just do separate mutagenesis reactions. So one day you could make the first mutation, the second day you can make the second one. But that's going to take a lot of time and energy. It would be a lot better if you could do them both at once. So if your mutations are at least 10 to 20 base pairs apart, all you need to do is add 2 different primers. And each of the primers is going to have its own mutation near the middle. Now I say they have to be 10 to 20 base pairs apart because if they're not that far apart, then they'll be overlapping and your reaction won't work. So they need to be at least a little bit far apart. They don't need to be completely opposite like I have shown in the picture here, but they need to have some spacing. Alright, so let's say you add your primers. Let's say here we have 2 mutations that we're making. One problem that can arise with this reaction is you can get a variety of different products. So you might create some plasmids that have only one mutation, others that have both, and then others that have, let's say, mutation B. So you can have plasmids with mutation A, with mutation B, or with both mutations. So just be ready going into that. We don't really have any good ways to prevent formation of the single mutated plasmids. You just have to go through it and then do your sequencing and see which of your plasmids is correct. But at any rate, it's the same reaction, you just add 2, or you add 4 primers instead of the 2. Now, some key differences here. So remember, we had 12 to 18 cycles in the past few reactions. We're going to bump that up to 30 cycles here. And that's because we're splitting our yield between these 3 different types of plasmids. So you add a few more cycles, that gives you more of your desired product. And there you go. Now, also, your extension time changes. So we're using 1 minute per kilobase. Now we're using 30 seconds. Now the reason we're doing that is because now we have 2 primers, so we have 2 polymerase reactions. So instead of a minute, you can do 30 seconds because you have both of them going at once. And finally, if you have a lot of mutations here, so there's kits out there that can do 5 mutations at a time, those will come with another enzyme that's called a ligase. And what the ligase does is it seals the nicks that are created during this reaction. So if you have 5 primers, you'll have 10 nicks, and you'll want to seal those with that ligase. Otherwise, the plasmid would not be very stable at all with all those different nicks in it. Okay, so you can use mutagenesis if you have a good idea of the exact mutations that you want to make for a specific reason. So I know this mutation will make the protein stronger, or I know this mutation will make it work quicker. It'll have a higher activity. If you have a protein that you don't know very much about, that hasn't really been studied before, so let's say you have this enzyme that does this reaction. It takes A and converts it into B. But that's all you know about it. If that's your situation, then the alternative to using mutagenesis, which is time-consuming and expensive, is to do what's called error-prone PCR. Now remember I mentioned that in PCR, if you add manganese, it decreases the proofreading ability of the DNA polymerase. So what that means is as you're doing the PCR and it's copying the genes, it's going to make mistakes. So you're going to be making then random mutations in your genes, in your PCR product. You have no way of anticipating what those will be, but you're going to generate this library of gene copies that have these flaws in them. And so you can see on the right, we start with the gene, and then about halfway down, we have all these different plasmids with all these different mutations in the GFP gene. Then the troubling or the time-consuming task becomes taking all those plasmids, putting them into separate E. coli, growing up those separate cultures, and directly assaying for protein activity. So if we have GFP and we want to make random mutations to make it brighter, that's the picture shown there, the fourth one from the top, where we actually grow the bacteria and then see which one of these cultures is the brightest. So you can see on the right there, the rightmost culture is the brightest. So that tells us that that mutation, whatever it might be, is the most effective at increasing the protein's activity. So we started off with, you can get literally hundreds of these mutations from error-prone PCR. Then you test them out, see which one works the best, and that's the mutation that you want to stick with. And you can use sequencing then to go back into that mutated plasmid and see where the mutation is, and then try to discern what the significance of that is. So, when you're setting up an error-prone PCR reaction, let's just go down the list on the left there. Use a normal PCR protocol. You can do mutagenesis or normal PCR. Doesn't really matter. You want to increase the magnesium concentration to about 200 millimolar. So you need more magnesium for this, and as shown in number 3, you want to also add more manganese. So manganese is the key ingredient here. Add that up to 25 millimolar, and your DNA polymerase will start making mistakes. So add your manganese. And what some people do is they also alter the nucleotide ratio. Now what this is going to do is it's going to limit the availability of certain nucleotides. So here they have 25 millimolar C and T, but only 5 millimolar G and A. So when a polymerase gets to that G or A, it's less likely to have it on hand, and it's going to probably put a C or T in its place. So the manganese and the altered nucleotide ratio work together to enhance the frequency of mutations. However, it's very important to note that you don't want a lot of mutations in the same PCR product. So we only increase manganese to 25 millimolar. If you went above that, you might get 10 mutations per gene, and that might make absolute crap. You might not get anything out of that at all. You want to keep it to about 1 mutation per gene. So stay pretty close to these numbers that we have shown here. And at the bottom there I have, you will need to optimize this reaction and control the number of mutations per PCR product. That's something you're just going to have to figure out on your own. You're going to have to try it, see if anything comes out. If it doesn't, alter some of the numbers you see here. Okay, and now that I've held your attention for about an hour, I'm going to show you the shortcut, the way to completely step around doing everything we talked about today. So, let's say you know the mutations that you want to do. All right, you know I have to make these 5 mutations. Instead of fooling around with mutagenesis, your other option is to just completely order a new gene. So there's several companies out there right now that will sell you completely or artificially synthesized genes. And these things can be from 100 base pairs all the way up to 100,000 base pairs. And only a quarter per base pair. So those costs can obviously add up, but we're looking at maybe hundreds of dollars for a new copy of a mutated gene. You're going to spend hundreds of dollars on a mutagenesis kit alone. So if you only need to mutate 1 gene, you might as well order it from a company. All the companies do is they take the gene sequence that you send them, so you can type it up in Word and email it. They take that gene sequence, and then they use this machine here. It's called a Mermaid 12 DNA synthesizer, and it uses a bead-based approach. So you take the bead, you flush it with the first base pair, so let's say it's a T. That T attaches to the bead, and then they do a few different things to it, and then they add the A, and then they add the C, and the G, and the T, and the C. And they've built this long DNA strand, And then at the end, they just cut it off the bead and ship it to you. And they're going to ship it to you already in a plasmid, ready to go. So, if you only need to do one mutation of a gene, go ahead and order it. But if you're going to do several mutations, you probably do still want to invest in mutagenesis. Alright, and that's about it for today. So hopefully I will see you guys on Monday, even though we have a storm scheduled for then as well. Again, if you have any questions about today's lecture, just let me know. Alright, also let me know if the sound quality on this thing isn't acceptable. Yeah. Alright, so enjoy the rest of your day off, and I'll see you later. Thanks.